Hi guys, welcome back to my YouTube channel. If you are new here, subscribe to be notified as soon as I upload a new solved past paper video with simplified explanations. Don't forget to like and comment. I'll be glad to answer any doubts. Alright, so let's get started. Figure 2.1 is a diagram of part of a DNA molecule. Draw a circle on figure 2.1 to identify one pair of bases. In DNA, the four nitrogenous bases are adenine, A, thymine, T, cytosine, C, and guanine, G. These bases pair up with each other. So here a base pair means two bases coupled together, which can be any one of these pairs. So let's say we can circle this one. Alright, so the percentage of T bases in the DNA of a species is 29%. Calculate the percentage of bases that would be base G in the DNA of the species. Now, some students might panic or get confused seeing a question requiring mathematical operations. However, I assure you, this is quite simple. Remember, in DNA, the four nitrogenous bases pair up in a selective order, A with T and C with G. Now, in the question it is specified to us that the percentage of T bases in the DNA of a species is 29%. Therefore, we can assume that the percentage of A bases in the DNA of the same species will be the same as in 29%. Now, if we add this 29 plus 29, it will add up to 58. So, the percentage remains for both C and G bases is 100 minus 58, which is 42. Since we're asked to calculate the percentage of bases that would be base G, we simply have to divide 42 by 2, giving us 21%. Now, for part C1. State the name given to a length of DNA that codes for a protein. The answer is gene. Remember that genes are specific sequences of DNA that contain the instructions for synthesizing proteins or functional RNA molecules. Right, so part C2, explain how proteins are made by a cell. If we were to briefly outline, it'd be somewhat like, proteins are made in a cell through the processes of transcription, DNA to mRNA, in the nucleus, and translation, mRNA to protein, at the ribosomes in the cytoplasm. However, notice that this is a 5-mark question, therefore we need to elaborate and give a detailed response. DNA containing the genetic information remains in the nucleus of the cell. During transcription, mRNA is synthesized from a gene within the nucleus. Once formed, mRNA exits the nucleus and travels to the cytoplasm where protein synthesis occurs. mRNA is transcribed from a specific gene located on the DNA molecule. This process, known as transcription. Ribosomes, cellular structures located in the cytoplasm, are responsible for assembling amino acids into proteins or polypeptide chains. This occurs during the process of translation. Once in the cytoplasm, mRNA attaches to a ribosome, serving as a template for protein synthesis. Ribosomes read the sequence of codons on mRNA during translation. Ribosomes facilitate the translation of the genetic information carried by mRNA into a specific sequence of amino acids. This assembly process results in the formation of a protein or polypeptide chain. The process of protein synthesis involving transcription and translation requires energy in the form of ATP. The order of amino acids in a protein is determined by the base sequence of mRNA. During translation, the sequence of codons on mRNA, which is complementary to the DNA sequence of the gene, specifies the arrangement of amino acids in the newly synthesized protein. This is the last part of question 2, right? So, DNA controls cell function by controlling the production of proteins. State 2 types of cell membrane proteins. Cell membranes consist of a variety of proteins that perform different functions essential for the proper functioning of cells. Carrier proteins bind to specific molecules, undergo a change in shape, and transport these molecules across the cell membrane. They are involved in facilitated diffusion and active transport processes. Another one would be receptor proteins. They are involved in signal transduction. They receive specific signals, such as hormones or neurotransmitters, from the cell's environment and transmit these signals into the cell, initiating cellular responses or triggering specific pathways. Some proteins embedded within the cell membrane function as enzymes. These enzymatic proteins catalyze specific chemical reactions at the cell membrane or facilitate metabolic pathways on the cell surface. Since they've just asked for 2, I will choose carrier proteins and receptor proteins. Alright, so here we reach the conclusion of the second part of the video. I will be posting the next part shortly. You can visit the entire playlist, and if you have trouble with selective questions, you can watch accordingly. I try to provide detailed but easy to understand explanations so that everyone can follow along and understand the concepts. Thank you for watching. Please like if you found the video helpful and subscribe for more.